DNA duplication, replication process. Initiation. The process begins at specific locations on the DNA molecule called origins of replication. Special proteins bind to these sites and cause the DNA to unwind, forming a replication fork. Unwinding. Helicase enzymes break the hydrogen bonds between the double strands of DNA, causing the DNA to unzip and create two single strands. Priming. Primase, an RNA polymerase, synthesizes a short RNA primer complementary to the DNA strand, providing a starting point for DNA synthesis. Elongation. DNA polymerase 3 adds new nucleotides complementary to the template strand, building new strands of DNA. This enzyme can only add nucleotides in the 5'3" direction. Leading and lagging strand synthesis. The leading strand is synthesized continuously, while the lagging strand is synthesized discontinuously in short segments called Okazaki fragments. Primer removal and replacement. DNA polymerase I removes the RNA primers and replaces them with DNA nucleotides. Ligation. DNA ligase joins the Okazaki fragments on the lagging strand, sealing the sugar phosphate backbone to create a continuous double-stranded DNA molecule. Termination. Replication ends when the entire DNA molecule has been copied.